Hello, good morning, students, and welcome to the Yusuf Biopoint. Students, today we shall start a most important topic, and it is very famous topic. That is, and you all know about this topic. DNA fingerprinting. It is the most important and usable topic. Understand? You know that at the time of making your Aadhaar card, the fingerprints are taken because fingerprints does not match to any other individuals. So means they all are different. And by that, so it is a unique identification things, and that's why in the same way the DNA is also used as unique identifications. In case of many crimes, you see about this one, and also you see when there is a dispute between two parents about a child. जब दो पेरेंट्स के बीच में किसी बच्चे के लिए झमेला उत्पन्न होता है, यानी कौन उसका सही पिता है, तो उस समय भी इसका उपयोग किया जाता है. So that is in the same way to identify, and it is the unique basis of identification. So DNA fingerprinting, you know that DNA is the genetic materials. It carries all the characters from the parents to offspring. So by using this DNA, and we see how much similarities found in the disputed persons and the persons about and in between the child and the disputed persons. यानी बच्चे और जो डिस्प्यूटेड व्यक्ति है उनके बीच में कितना यह है, या कोई भी क्राइम्स हुआ तो वहां से हमें कोई फिंगर रिपोर्ट मिलता है, कोई भी सैंपल मिलता है, तो उससे और ये डीएनए फिंगरप्रिंटिंग से हम पता लगा लेते हैं. समझे? तो ये व्हाट इज द डीएनए फिंगरप्रिंट. The idea about DNA fingerprinting was given by Alexander. We know that in the DNA sequence, about 99% similarities are found in all individuals. About so much percent, 99.9% similarities are found in about all the humans. Understand? Now we are telling about humans, so use only the differentiation is 0.1%. This is the differentiation, and our duty is to find the sequence of this different type of segments of DNA and compare with others. यानी हम इसी 1% से हम डीएनए फ्रैगमेंट से निकालते हैं और इसी 1% के डिफरेंशिएशन से अन्य व्यक्ति के डीएनए सीक्वेंस से हम जो पता लगाते हैं. दैट इज बिकॉज 99.9% लगभग तो सेम होते हैं. तो जो डिफरेंशिएशन है उसी बेस पर हम किसी को आइडेंटिफाई कर सकते हैं. नाउ यू सी दैट Alexander divide the DNA sequence into one is a long sequence and next one is short. So long is called bulk DNA and short one is called sat DNA, means satellite. This satellite DNA, understand? This satellite DNA is of two types. One is microsatellite and next one is mini satellite. One is called microsatellite and next one is called mini satellite. He differentiated he divided it on the following features. One is AT rich और GC rich. उसमें AT ज्यादा पाया जाता है या GC ज्यादा पाया जाता है, एडेनिन थायमिन या गुआनिन साइटोसिन. मीन्स द रेशियो बिटवीन AT and GC. What is the ratio between AT and GC? On that basis and that. Next one. Length of the फ्रैगमेंट्स जो है उसका लेंथ क्या है, फ्रैगमेंट्स का लेंथ क्या है, लेंथ ऑफ द फ्रैगमेंट. थर्ड, नंबर ऑफ टाइम ऑफ रिपीटिंग सीक्वेंसेस. जो रिपीटिंग सीक्वेंस है उनकी संख्या कितनी है, एक ही जैसे मतलब सीक्वेंस कितनी दफा है, यानी कहना मतलब उनकी कितने एक ही जैसा जो होता है उसका कॉपी कहलाता है. इसका मतलब कि एक तरह के सीक्वेंस कितने हैं. व्हाट इज द नंबर ऑफ सीक्वेंस व्हिच आर रिपीटेड, मींस व्हाट इज द नंबर ऑफ सीक्वेंस व्हिच आर रिपीटेड. जो नंबर ऑफ सीक्वेंस इज रिपीटेड इसको हम लोग कॉपी नंबर भी कहते हैं. तो सो ही on this basis he divides the satellites in microsatellites and mini satellites, understand. And microsatellite is used as VNTR. Microsatellite is used as VNTR. Now what is VNTR, you see. Now the Alex Zafari used the satellite DNA as probe that show high degree of polymorphism. It was called PMR. Polymorphism means that is different in the sequence of DNA. We know that in the DNA A, T, G, C, A, T, G, G. So such like bases are found. If they, by means of mutation, if T is changed in place of C, there by mutation some changes occurs, that is called polymorphism. Means the differentiations occur in the sequence of DNA or in the basis of DNA due to mutation. So and when the due to mutations that differentiation occur, it means the sequence or the fragments, structure of fragments will change. So it is not called, it was called VNTR, means variable number of tandem repeated. Means that now you see what is a tandem. So tandem is short sequence of DNA that repeat head to tail fashion. Means short, this is a short, and now this is head, this is tail. How such type of sequence are found? In the same manner, Means if the sequences are same, then we will call that it is repeat. जब एक ही तरह की चीज हम कई पाए तो हम कहते हैं कि दैट इज रिपीटेड. तो सो शॉर्ट सीक्वेंस जिनका फैशन हेड टू टेल एक ही जैसा है, वह कितनी बार, हाउ मेनी टाइम्स it is found, that is called tandems. So variable number of tandems, so such type of tandems in found in how many numbers, that is called VNTR. Now what are the steps and what are the method of this DNA fingerprinting? First we isolate the DNA, means first we make the sample of DNA, and for that we take the samples. The samples may be blood, maybe hair, or maybe sperm or any parts of the body. For that we can take to gain DNA from any parts of that. And after the isolation, we shall get the DNA fragments in the long bases like that. And we know that DNA is found in double helicals, so it is found that. So now after that we shall digestion, or now we separate is like that. This is the two DNA. After by using the endonuclease enzymes, we separate it, and now this separates their joints. So means digestion or fragmentation by isolated DNA by the enzymes endonuclease एंजाइम के सहायता से हम उसे छोटे छोटे खंडों में फ्रेगमेंट्स बांट देते हैं. And now about this method we will study in the biotechnology, which will be your next chapter. So in all the chapters, all the topics which you are seeing here, about all you will study in the biotechnology. There is a short description. So after making fragmentation, we will separate the fragments by gel electrophoresis. We'll separate this by gel electrophoresis. Now, what is now the gel electrophoresis is a technique used to separate DNA fragments according to their size and charge. Means we take here is a medium, that is agarose medium. It is electrical connections, and as your DNA fragments. And we know that DNA is a negative charge. DNA is a negative charge. So on electrolysis, it will deposit it on the positive charge plus. So that method is known as electrophoresis. The gel we use, that agarose gel. So after that, after the separating days, transfer this on the blotting this on the nitrocellulose sheet. Means after that, we shall, it is the sheet, with that we shall take on that this pattern on the nitrocellular or nylon sheet. Now the hybridization by VNTR. Now how the VNTR method we shall take. Suppose that A is the person, A is the child, two parents B and C. A child, B and C, two persons who claim that A is their child. B claim for A and C also claim for A. A मान लो कि बच्चा है, दो व्यक्ति हैं जो दोनों क्लेम करते हैं कि यह ए उनका बच्चा है, बी भी क्लेम करता है और सी भी क्लेम करता है कि दैट इज ए देयर चाइल्ड. नाउ दिस प्रोसेस will be done for to identify. Now we take the sample of B and make the ventil. Suppose the DNA which I have found A1, A2, A3, A4, A5 like that. From the C, the fragments are found X1, X2, X3, X4, X5 like that. And the A, this also we can take, and we shall see how much more similarity is found with A, B or C. हम A का भी हम सैंपल लेंगे, B का भी C का सैंपल लेंगे और सबों का इस मेथड से फिंगरप्रिंटिंग किया जाएगा, एनालिसिस किया जाएगा. अब जो उसे डीएनए सीक्वेंस मिलेंगे, अब डीएनए मान लो बी के सीक्वेंस है, ये सी के सीक्वेंस है और ये बच्चे ए का सीक्वेंस है. अब देखेंगे किसका जो है किसके साथ ज्यादा सिमिलरिटी होती है. जिसके साथ ज्यादा सिमिलरिटी होती है, ज्यादा समानता होगी, सपोज ए का सी के साथ है तो मान लो इसका भी x1, x2, x3, x4 इस तरह से पाए गए लेंथ. तो अब ये जो पाए गए हैं समझे, तो अब जिसकी समानता जिससे ज्यादा होगी वह बच्चा उसी का है, समझे. सो बाय दिस वे वी कैन आइडेंटिफाई दैट हु इज द एक्चुअल पेरेंट्स ऑफ सच disputed child. And so by this way, or any case, from any matter, whatever the sample we get, and by the comparing the DNA fingerprintings and other the suspected persons, we can identify and then we can decide that which one is responsible for that